Good morning, everyone. My name is Stephanie Fonseca. I am a metallurgical engineering student. Today, I would like to talk about a research article focused on the leaching of sulfur minerals containing gold. To begin with, I would like to comment that this article is from the researchers Iglesias and Carranza from the Department of Chemical Engineering of the University of Sevilla. The name of the article is Treatment of a Cool Bearing Arsenopyrite Concentrate by Ferric Sulfate Leaching. Therefore, in this work, with a view to improving the reaction kinetics, the leaching with the ferric sulfate of an arsenopyrite concentrate is studied, exemplifying a direct deoxidation process, where the chemical process are carried out with the attack of sulfate ion. Ferric and the biological part is attributed to the oxidation of the ferrous iron processes. The result obtained after the process are satisfactory, where the kinetics of ferric sulfate leaching is improved after the use of silver as a catalyst. At moderately high temperatures, the study focuses on the optimization of the chemical part, since the biological one has been tested on many occasions. On the left, you can see the biolation bioreactor. Refractory gold minerals are those that, after extraction of gold by conventional cyanidation, have a mineral grade of less than 80%, so they must treated before cyanidation so that the gold comes into contact with cyanide ions and decompose species that deplete cyanide and avoid available oxygen to dissolve gold. Note that bacterial oxidation as a pretreatment is a process in which the leaching of sulfur minerals is catalyzed by microorganisms. These bacteria oxidize sulfur and ferrous iron sulfate and ferric iron. Exclusively Thiobacillus ferroxidans is used. The concentrate was leached with sodium cyanide to test the effectiveness of the oxidative pretreatment. The experiments were carried out in cylindrical reactors of 5 liters capacity, equipped with baffles and a glass tube to supply compressed air. In addition, they were equipped with a cap with pores for measurement, pH, and sampling and minerals addition. The test was carried out at room temperature, adjusting the pH. Initially, liquid samples of gold, silver, and arsenic were taken. The gold was concentrated by extraction with methyl trioxyl ammonium chloride. Carbon disulfide was used to dissolve the sulfur produced. The leaching tests were carried out with silver as a catalyst, and an electrode was used to control the pH. If we take a look at the slide, we can see a diagram and a table that refer of the chemical composition of the concentrate and the particle size distribution. Note that the gold contained is 14 ppm, silver and iron are 88 ppm and 4.6%. In the graph, we note that the particle size die 5T is 86 micrometers and the value D80 is 136 micrometers. This being the main particle size. Finally, the experiment that part a table is obtained with the identification of phases present to the concentrate. Is using different techniques where arsenopyrite is the most abundant mineral. With this study, it is allowed to conclude that the temperature are important since less than 50 degrees centigrade net chemical control of the process given is a regard to the leaching reaction to 17 Celsius degrees together with the action of silver microbial ions, the activity of the chemical effect leaching with silver catalyzed ferric sulfate has also demonstrated and the biological effect bioxidation of ferrous iron is a used known process which allows the regeneration of ferric sulfate. My point of view of the research is favorable because this alternative for the extraction of precious metals such as a gold with cost reduced and reagents to be used through chemical and biological routes helps to protect the environment. As we know that the gold extraction industry is very transgressor of the environmental part. From the psychochemical point of view, the point of variation of the pH is important to maintain the microbial activity as well as the temperature variation of the where it is possible to overcome to initial problems of kinetics and speed of reaction with many more benefits. Thanks for listening me.